Hello, my name is SERCAN. I am happy to share our recent work on an mRNA vaccine candidate for Crimean Congo hemorrhagic fever virus, or CCHFV. CCHFV is a tick-borne virus with a high fatality rate, up to 40%. It is mostly seen in Africa, Asia, the Middle East, and parts of Europe. Because of climate change, ticks carrying the virus are spreading to new areas. There is no approved vaccine for this virus yet. The World Health Organization lists CCHFV as a priority virus that urgently needs vaccine research. So, there is a clear need for a safe and effective vaccine. In our study, we designed a pseudouridine-modified mRNA vaccine that encodes the nucleoprotein, NP, of CCHFV. We combined the mRNA with PLGA nanoparticles and tested it in mice. We focused on both humoral and cellular immune responses, especially looking at the levels of inflammatory cytokines. Here you can see the mRNA production workflow. We used a DNA template with the CCHFV-NP gene and a T7 promoter to synthesize the mRNA. To improve stability and translation, we added 5' and 3' UTRs, a poly-A tail, and used pseudouridine instead of normal uridine. These features help the mRNA last longer and work better. We checked the mRNA quality using agarose gel and nanodrop to make sure it was pure and intact. Then we confirmed mRNA expression in vitro and moved on to mouse immunization studies using both naked and PLGA-formulated mRNA. We also did challenge experiments, but those results are not shown on the poster. The red line in the graphical overview on the poster indicate the immunological analyses we did. We present these results here. In our results, we used two mouse models, BALB-C mice with normal immune systems and C57BL6 mice with transiently immune suppression. I will focus on the BALB-C model here. We divided 24 female mice into 6 groups and gave them 3 vaccinations, each 14 days apart. Figure 1 shows that vaccination with IVT-NP, pseudouridine mRNA, triggered a strong antibody response. We also checked the PLGA-formulated mRNA using dynamic light scattering, confirming particles around 100 nanometers with a negative charge. After immunizations, blood samples were collected and tested by ELISA. The highest IgM response was seen 14 days after the first dose, especially in the IVT-NP pseudouridine mRNA group. IgG levels increased after each vaccination, reaching the highest in the pseudouridine-modified mRNA group. The unmodified mRNA group had the lowest IgG response. Antibody avidity, strength of binding, also increased over time, again highest in the pseudouridine-modified group. Western blot confirmed that antibodies from vaccinated mice recognized both recombinant NP and NP expressed from IVT-NP pseudouridine mRNA transfected cells. In summary, IVT-NP pseudouridine mRNA, both naked and PLGA formulated, induced a strong and high-quality antibody response. Figure 2 shows the cellular immune responses induced by our vaccine. We collected spleen and lymph node cells from immunized mice and stimulated them in vitro with NP and other antigens containing NP. Flow cytometry results showed that CON-A caused the highest cell proliferation as expected. Importantly, recombinant NP, RNP, and lysates from IVT-NP, pseudouridine, mRNA transfected cells also caused strong and specific T-cell responses. We measured these responses using proliferation and division indices, confirming significant T-cell activation. We also used BRDU labeling to confirm this. T-cell proliferation increased with higher antigen doses, and we observed dense clusters of cells growing in culture. Next, we checked cytokine gene expression. Compared to controls, levels of IFN gamma, IL-12, TNF-alpha, IL-2, and IL-15 mRNA were much higher. These results show that our vaccine triggers a strong and specific Th1-type cellular immune response. These results are from transiently immunosuppressed C57BL6 mice. Figure 3, 4, 5, 6. Figure 3 summarizes the NP-specific antibody responses before and after the viral challenge. We measured IgM and IgG levels in mice on days -1, +2, and +14 relative to the challenge. High IgG levels were observed both before and after the challenge in groups immunized with IVT-NP pseudouridine mRNA, either naked or formulated with PLGA. Mice that were fully protected had higher antibody levels, while partially protected groups had lower levels. The same pattern was seen for IgM responses. Surviving mice showed strong antibody levels after challenge. These results were confirmed using both in-house and commercial NP-based IgG tests. Overall, these findings support that our vaccine induces a strong humoral immune response that helps protect against the virus. Figure 4 shows the avidity of NP-specific IgG antibodies in immunized mice. Before the challenge, the highest avidity was observed in the IVT-NP pseudouridine mRNA PLGA and naked IVT-NP pseudouridine mRNA groups. These were also the groups with full protection. In contrast, groups with partial or no protection showed lower avidity before challenge. After the challenge, surviving immunized mice in all groups produced high avidity antibodies, with the highest values still in the pseudouridine modified mRNA groups. We also followed avidity changes over time in the same mice and saw a clear increase after each exposure. These results suggest that our vaccine not only induces strong antibody levels, but also promotes affinity maturation, which is important for long-term protection. Figure 5 shows the cellular immune profile after viral challenge. 2 days after the challenge, we analyzed spleen and liver tissues by flow cytometry to measure CD4 and CD8 T cells. We observed a clear increase, up to 30% in CD8 T cells, especially in groups with 100% survival. This indicates a strong cytotoxic T cell response, which likely helped clear the virus and provided protection. These findings highlight the key role of CD8 T cells in the protective immunity induced by our mRNA vaccine in immunized mice. Figure 6 shows cytokine gene expression in spleen and liver cells measured 2 days after infection. In immunized groups with higher survival, we observed strong increases in key cytokines such as IFN-gamma, TNF-alpha, IL-10, IL-12, and IL-2. The IVT-NP pseudouridine mRNA groups also showed higher levels of IL-15, GM-CSF, and IL-4. There were slight increases in IL-10, IL-6, and IL-5 in these groups as well. Interestingly, IL-1 beta increased only in inactive CCHFV immunized livers, and IL-17A was detected only in the spleen. In conclusion, our pseudouridine-modified mRNA vaccine encoding CCHFV-NP successfully triggered both humoral and cellular immune responses in immunized mice. High levels of IFN-gamma, TNF-alpha, IL-2, and IL-12 suggest a strong cell-mediated response, which is important for clearing the virus. The increase in CD8 plus T cells in the liver and spleen was linked to protection, showing the importance of cytotoxic T cells in mRNA vaccine responses. Our results show that pseudouridine-modified mRNA vaccines can create strong and targeted immune responses against dangerous viruses like CCHFV.